Okay, so hello everyone and welcome to the Genetics and Genomics virtual event. My name is Manuel Kaulich. I'm a group leader at the Goethe University in Frankfurt, and I'm also the Chief Scientific Officer of Vivlion, a recent spin-off of the Goethe University. I will be presenting circular synthesized CRISPR-Cas gRNAs for functional interrogations in single and multiplex formats for you today. Please feel free to submit questions during my presentation, and I will follow up with you after the presentation via email. So first of all, I would also like to thank LabRoots for giving us the opportunity to present ourselves here in this audience. And for those that would like to get more information, please visit us on the web or contact us directly. So let's get started. So I'd like to take a few first words and just introduce ourselves briefly. We are a very recent spin-off of the Goethe University with Ivan Dikic as our executive, Kerstin Koch as operating officer, and myself as chief scientific officer. Our main mission in Vivlion is to provide single and multiplex low and high diversity CRISPR-Cas or RNAi shRNA libraries, and I will briefly introduce to you today the technology that we are using and also what we can do with these reagents and what you can do actually with these reagents. So this brings me already to a few introductory words. When we talk about CRISPR applications, of course, we mostly mean, as you will see on the left-hand side of the slide, talk about the wild-type Cas9, and in this case, the SP Cas9, where basically a guide will be delivered to a single cell and then induce a double-strand break, which is what we today understand as conventional gene editing to perform single or unbiased gene screens. But of course, the technology also enables much more than that. When combining, for example, with a inactivated Cas9 enzyme or a dead enzyme, we are able to perform base editing today, RNA targeting. We can change chromatin topology or even visualize chromatin. We can change the epigenome. And of course, by that, I also include CRISPR-A and CRISPR-I for the regulation of specific transcripts or genes. So when we talk about generating reagents for single or even screening purposes with CRISPR-Cas, there are a few caveats that I would like to point out to you, especially when it comes to the generation of libraries for gRNAs. Most of us, if we do generate libraries today, we sort of depend on PCR and conventional cloning techniques. And that's not surprising, because when we want to make a highly diverse reagent, like for example a genome-wide library, then the material that we get off the synthesizer is fairly limited. And what that means is, what that results to is actually that we have to PCR amplify our product. And then this is a natural step, and what that means is that we introduce sort of a PCR bias into the distribution of our sequences. So now that we have our PCR product, we need to digest it with restriction enzymes, or we apply or do homology-based cloning. Nevertheless, what we have to do is we have to take our PCR product and put it into somewhat open chromatin or plasmid. And this is the second problem, which is basically cloning artifacts. And when we now close those by, as I said, homology cloning or by ligases like T4, for example, and then transform them into bacteria, we will get artifacts which either have no guide RNA or are truncated versions of our plasmid. And this is then what we basically call our plasmid pool or our final library, which will then, of course, also be sequenced. And this, by sequencing, we will only look at the part that we are really interested in. So we will not sequence, for example, all the artifacts that are in the plasmid pool as well. So by saying that, what I'm trying to say is that really conventional technologies as of today depend on PCR amplification steps. They do require restriction enzymes because we have to open our chromatin. And as I said, they need conventional cloning steps. and until we get our plasmid pool, of which we then later generate our lentiviral pool for the actual experiment. So, as I already alluded to, the PCR and cloning step can really be a bottleneck here, because when we take, for example, reagents that are published and that are out there from distributors, then and we really just look at the quality, the diversity and distribution of these libraries, what we see is that, for example, several of those, the orange, the yellow, and the blue, especially here, that they have a certain distribution, which is different than from, for example, a reagent that we have generated with our technology that I will allude to you in a little bit, and that refers to the FDA library. So what you see on that graph is what we call a Lorentz plot, and that basically visualizes the distribution or the evenness, the uniformity of a library. And the more flat this curve is, desto better or evenly is this reagent distributed. Furthermore, what is highlighted here is also what we then calculate the area under the curve, which is for the FDA, if you look at this, is 0.57. So the area under this curve is 0.57. And the closer it gets to 0.5, the more even it is distributed. And you can see that the previously published reagents, they are less good distributed than, for example, a reagent that we have generated. The other thing, and despite distribution disadvantages due to PCR, I already mentioned are cloning artifacts, and this is shown here exemplified by basically a pooled reaction, a pooled ligation reaction using conventional methods. And what we observed is that only roughly about half of the clones that we then picked from a plate and Sanger sequenced and verified also by the digest that you can see here were correct. And this is demonstrated here by the lower band after restriction digest. While several clones, for example, in lane 2 or in lane 9 or even lane 17 showed complete artifacts, which are based due to the cloning procedure. And those will always be in your plasmid pool and may or may not generate virus and thereby affect your library. So there are some caveats by using PCR and conventional cloning, and I would like to share with you a protocol that we have come up with that completely circumvents the need for PCR and cloning. And this is exemplified here in an easy scheme. So what we can do is we can take any plasmid with or without Cas9, RNAi expressing plasmid, shRNA expressing plasmid, or even a cDNA encoding plasmid, and convert this into a single-stranded piece of DNA. So what we do is we take that double-strand character away, we turn it into a single strand, and then we take this single strand, which is now this gray circle. What we do is we anneal oligonucleotides to it with 5' and 3' homology, and the triangle in that orange arrow indicates the new sequence that we would like to integrate. So now we don't do PCR, but instead what we do is we extend this primer towards its own tail, thereby generating a heteroduplex of, in gray, the old template strand, and in orange, the newly synthesized strand. Yeah? So the trick here really is that this gray template strand is chemically modified. And it's chemically modified in a way that enables us, during the transformation, that bacteria will recognize this modification and then will degrade this gray strand and only amplify the newly synthesized orange strand, and thereby basically produce the final library in a pooled manner. So again, this protocol is fairly rapid. It only takes about 2 hours, and it does not involve any PCR steps, no restriction enzymes. We don't have conventional cloning. Of course, we have bacterial amplification, but not ligation-mediated cloning. And it's very, very, very rapid, and it is versatile, as it does not is not limited to only CRISPR-Cas, but as I said before, it can also be used for shRNA, for example, or cDNA. So does this really work? It's shown on the next slide. And first, what we tested is if we can use previously published CRISPR-Cas plasmids and tested if we can convert them into single-strand DNA. And this is shown here. We used as a positive control, we used pcDNA3, and I hope you appreciate that the pcDNA3 double-strand character runs as a doublet or triplet band, and then the single-stranded form migrates much faster as a single band. Likewise, two conventionally used CRISPR-Cas plasmids, pLentiGuide and pLentiCRISPR version 2, in double strand as well as in single strand character, demonstrating that we really can efficiently generate single strand DNA of those plasmids. We then went on and performed a test synthesis on these two plasmids, shown here with a pool of 6 oligonucleotides. And these oligonucleotides encoded for gRNA sequences targeting GFP. Yeah, so those are mini pools with 6 guides. We performed the synthesis, and the synthesis product is shown here on the gel. We then purify this and transform it into bacteria, as I alluded to you before. So then what we did initially is we tested individual clones if they contained the correct sequence. And this is shown on the bar graph, on the first bar graph. And around 80% of the clones were correct, while 20% roughly contained the wild-type sequence of our template plasmid. So then we basically went a step back and we introduced a dummy gRNA sequence encoding for an ISC1 restriction enzyme site to get rid of any wild-type remaining remnants in our pool. And when we then basically take our plasmid pool and clean it up with this restriction enzyme, then I hope you appreciate, and this is the second bar, that any of those clones that we now look at is now the correctly mutated one, and there's no wild-type left. On this pool then, which was only 6 gRNAs or GFP targeting gRNAs, we then performed next generation sequencing to basically see what is the distribution in this library and also the distribution. And I hope this is on the very right side shown here. This is NGS data, and what we are plotting here are the 6 individual gRNA sequences against their copy number in log10 ratios. And I hope you appreciate that each of them has been identified nearly uniformly. So there was no apparent bias towards one or the other sequence. So that was very interesting and then a positive result. And then we went on and took those sequences, produced lentivirus of them, and applied them to cells expressing GFP. And this is shown here with an T7 endonuclease assay demonstrating the functionality of these constructs, either in untransformed cells, so this would be the first lane with the minus, or cells transduced with virus encoding for a non-human target sequence as well. There was, as you can see, there was no editing. However, when we added in a single GFP targeting guide, we could identify substantial editing, and this could be further enhanced by using the pool that we generated before. Also demonstrate, and we even observed multiple bands popping up, really indicating that all of these guides potentially have cut it and basically shredded the GFP locus. So this was for us functional evidence on the genomic DNA, which then of course also translated to the protein level. When we then moved these same cells to the FACS, what we observed is basically in non-human target control cells, 100% of these cells were GFP positive, while in the GFP pool transduced cells, we had a dose-dependent reduction in GFP over time, demonstrating now that the reagents were functional on genomic DNA, which then of course also translated to the protein level. So this was great because we now had a technology at hand that would generate us these reagents, and we didn't know at the time if it would be suitable also for generating libraries. So what we did is we tested this. And the way we tested it is by mimicking library diversities using randomized oligonucleotides in primer sequences. And this is shown here on the left side. We designed oligonucleotides with 4 ends or until 9 ends, and thereby basically mimicking a diversity of 265 until 260,000 different oligonucleotides. So the beauty here is that these individual oligonucleotides can be purchased from any oligonucleotide provider, and basically they guarantee you the full diversity. And so we ordered those 6 oligonucleotides, performed the synthesis, and also sequenced then these reagents, and this is shown here on the right side. Again, this is the Lorentz plot showing uniformity of these reagents, and I hope you appreciate that the reagents were full, complete, and were very nicely distributed. So now what we did also test at the time was if this is only due to randomized nucleotide libraries, or also to really encoding libraries. And so what we did is we also generated a library targeting all human deubiquitinating enzymes, which is shown here on the right hand side, on the bottom in this greenish color. Generated this library, and it had an area under the curve of 0.6, so demonstrating very nicely a distribution of this library. So, but what was the most important conclusion for us here is that the technology that we have uncouples sequence diversity from sequence distribution, which is independent of different from conventional cloning, where the more reagents you would like to clone simultaneously, the worse basically the quality of your final product is. So with this technology, this is not the case. It is basically doesn't matter if you have a single sequence that you would like to integrate or 100,000 sequences, the quality of this reagent will be the same. So by this, from this slide, I would like to conclude that the 3CS, so we call it 3CS because what we do with this technology is we generate covalently closed circular synthesized libraries, and we can do this in any order of complexity, and that these reagents are unbiased. Then we went on and took the stub library that I just mentioned, and what we did is we generated, of course, lentiviral particles of it, and we performed a dropout screen with these particles, and the scheme, how we did it is shown here. We applied an MOI of 0.2 and an experimental coverage of 1000. We used H-third immortalized RPE1 cells, and we waited 2 days after the transduction to take a day 0, our control time point, and then applied a puromycin selection pressure, waited another 11 days, and yet another 10 days. So we have 2 time points here, day 11 and day 21, at which we then harvested cells and also extracted the genomic DNA. We went on for sequencing, and first of all, what we did, of course, we compared our biological replicates, which is shown here, how they correlated, and basically for day 0 on the left side, for day 11 in the middle, and for day 21 on the right side. Hope you appreciate that our R-squares and Spearman correlations are very high, above 0.9, and demonstrating a very high experimental reproducibility with these reagents. And of course, high reproducibility also led us then to perform a dropout analysis or hit calling, and this is shown here on this slide. For each day, we were then able to identify deubiquitinases that either enforce a positive fitness effect or a negative fitness effect. And this was interesting because I put your attention to day 11 and day 12. USP28 was always a very positive fit, which was already known from the literature, but for example, BRCC3 or USP9Y were not previously known to enforce a positive effect, so there was novelty here. But also we could confirm negative effects of DAPs, like for example COP-S6, USP7 or PSMD14. Interestingly, since we took 2 time points, we were also able to identify phenotypes that may change over time, and this is demonstrated here on the right hand side. For example, USP28 was strong at day 11, but was even stronger at day 21, which why it was a really strong hit in terms when it changed to phenotype on the right hand side. Just to mention a few, and of course non-human target sequences were always sort of like in the border of being significant, as they were our controls. To sum up this experiment, we then of course identified enriched and also depleted deubiquitinases. That is shown here. We were then able to use this information and apply our technology to generate shRNA reagents. This is shown on the right side, that we then used in an independent validation approach to basically confirm or validate the phenotypes that we had identified with our reagents before. And we used a luciferase as a negative control and the PLK1 targeting as hRNA as positive control for dropout. And I hope you appreciate that we could re-identify USP28, BRCC3 as inducing a positive effect, while USP7 and COP-S6 were identified or validated to induce a negative effect. So what this now enables us basically to do is positive and negative screening, and of course we can use the technology to generate the shRNA or gRNA validation reagents. To sum this real quick up, of course, on an educational purpose, there are different levels in terms of what is needed, what is equipment and needed, and what the size of these experiments are. And I would just like to walk through with you with this real quick. So of course, easy phenotypes are, for example, survival or essentiality, which basically translates into a dropout or an enrichment screen. The library format in this case is mostly pooled, and the instrumentation needed would be next generation sequencing. For medium level screens, of course, we are also considering this an on and off phenotype that we can sort. For example, if we have a fluorescent reporter construct, the library format would still be pooled in this case, but of course the instrumentation changes as we now also need to include a fact sorter as well as an NGS. Advanced level of screening are really large single or image-based phenotypes or screens in which the library, as I said, is arrayed, so each well has a different knockout. That of course requires high content instrumentation with imaging or plate handling. So now having basically this technology established in the lab, we wondered how far can we go with this technology. And by saying this, I mean, can we go truly genome-wide? I mean, before people have generated genome-wide libraries, although one has to be careful with this term, as it only means coding-wide. And genome-wide is a different meaning as coding-wide. And what we mean here is really truly genome-wide. So basically generate a reagent that not only targets the coding but also the non-coding region of the human genome, which of course is the vast majority of the sequences. To do that, we took advantage of the literature, and we where people have identified nucleotide preferences of SP-Cas9 in gRNA sequences. We took this information and we translated it into UPEC code, and this is shown here. And by doing so, we basically generated a partially randomized oligonucleotide that is specific to target highly active SP-Cas9 target sites in the human genome. And if we take this information and we translate it into UPEC, then this is the sequence in orange here that you will come up with. It has an estimated diversity of 7.3 to the power of 10 trillion sequences, so it's very, very diverse. But nevertheless, by having this oligonucleotide sequenced by an oligonucleotide provider, you will still get all these different primers. So what we did is we did apply the primer to the two plasmids, two lentiviral CRISPR-Cas plasmids that I mentioned to you before, p-lenti-guide and p-lenti-CRISPR version 2. We performed the reaction, and then we made the library and also sequenced the library. So of course it's not possible to sequence this library in its full diversity, but we minority sequenced it. And when we did that, we looked at how frequent are individual nucleotides at each gRNA position. This is shown here on the right hand side. And as we would have expected at position 1 next to the PAM, we have an enrichment for A and G, while C and T are disfavored. And this then translates into a UPEC code of R. And now I hope hope you appreciate that if we walk through all of these sequences, then we re-identified the same UPEC code as we had originally put into the synthesis, sort of demonstrating that we have generated at least a sufficient diversity in this library. In theory, this library targets 20 million SP-Cas9 target sites in the human genome. So this is about 20 orders of magnitude, or even more higher than, even 200 times higher than what has been done before. We then went on and made a lot of virus of this library to really make sure that we have at least a certain degree of library representation. And in this case, we did 8 different reactions that we pooled. And we synthesis pooled, 8 big libraries that we pooled. We performed several transfections to make lentiviral particles. We then collected all of these together and generated roughly 500 milliliter with a titer of 10 to 7, or 10 to 6, excuse me, 55 milliliter with a titer of 10 to 7. We then took this and applied the same lentiviral pool to a very strong selection, positive selection, again using our RPE1 cells with an MOI of 1. And the selection was essentially doxorubicin resistance, from which we knew before that 1 micromolar of this drug would sufficiently kill these cells within 3 to 4 days. As highlighted here on the right hand side, so we waited 3 weeks after doxorubicin, or in presence of doxorubicin, and we considered every cell that would still be alive on the plate to have some sort of resistance mechanism in place. We then harvested these cells and we sequenced the gRNAs, or the sequences that were contained in there. We did this in biological triplicates, and we were able to identify an overlap of roughly 4,200 sequences. While the individual experimental overlap was fairly low, nevertheless we were able to identify experimental overlap. And of course now the question is, how is this knowledge that we generated here somewhat applicable? And we say that with this reagent you will be able to identify novel coding and non-coding therapeutic targets, for example, and in this case the target would be doxorubicin resistance. We then made a new library, and this really is the power of this technology. It's very iterative, so we can generate these reagents very rapid. So we took now these 4,200 sequences, made a new library, and performed the screening again. And this time we applied a gold standard or established experimental conditions of MOI 0.2 and also experimental coverage of 1,000. And waited again 3 weeks in the presence of doxorubicin. And here we can see that again we identified roughly 2,800 sequences that were able to be re-identified in the screen, of which about 800 were again enriched by more than 2-fold. And those we now consider basically experimentally validated. So now the question is here, of course, where do these sequences reside in the human genome? And we did look first for the coding targets, and we did identify several of them, also in the dependent on mismatch. Of course, as pCas9 is not a clean enzyme, so we have to take at least 2 off targets here into account, or 2 mismatches at least. And by doing so, we did identify several genes, and among them was one particular gene that was interesting to us because it had previously been published to be a doxorubicin resistance marker, and this gene is called cis-LTR2 on the left side. And when this is why is it so interesting? Well, it's also interesting because there are 2 chemical compounds available targeting this gene or protein, and when we take those compounds, they are shown here on the right side and called bi-cis-LT2 or bi-U9773. We take those compounds and we titrate them into doxorubicin, into increasing concentrations of doxorubicin. What we could do is we could basically reverse the toxicity of doxorubicin, chemically validating our findings here. However, these were only the coding targets, and of course more interesting were the non-coding ones. And when we now look at non-protein codings, what we could see is, I just click on the next slide as well, so we have the summary here. We did see, of course, now on the left side, we did see, of course, also an enrichment in hits while allowing mismatches, which is not too surprising. However, overall we identified a lot of predicted promoters of genes, long non-coding RNAs, antisense, pseudogenes, CTCF binding sites, which had been previously reported to be important in this context, but also transcription factor binding sites and predicted enhancers. However, I would like to draw your attention to the very lowest part, which is the biggest part of this pie chart. Roughly 52% of all of the sequences that we re-identified, we were unable to identify any, and so we could map them, but there was no annotation available. So we know that the sequence is doing something with respect to doxorubicin resistance, but we don't know what the function is at this particular site. And this is, of course, ongoing in the labs to basically validate them individually and see what is going on there. So now this was what we call the TGV library, the truly genome-wide library, and of course it is very, very big and probably too big to handle. So what we did is we generated an optimized version of this, and by saying this, what I mean is that we used a truncated version, and this has been shown before. Truncated gRNAs down to 17 nucleotides have been reported to be similarly active than guides with 20 nucleotides in length, and this is shown here. So what we do basically is we deleted the first 3 nucleotides, and thereby not so much affecting the 20 million target sets in the human genome, but dramatically reducing the diversity of this reagent, and now making it very feasible to actually screen these reagents in suspension or even in adherent cells. We generated this reagent on P-lenti guide, P-lenti CRISPR version 2 with Puro, as well as with a GFP marker, and those reagents are partially again also quality verified, that we do by electrophoresis and also by NGS, and this is showing you here an example for the P-lenti CRISPR version 2 library, where we basically confirm by NGS again the UPAC code that we had originally put into, demonstrating that we have these reagents at hand and we are sharing them. And if you would like to read more about this, then I would I kindly ask you to also to look at the publication that we are referring to here. So now we can very rapidly and with high confidence generate high quality unbiased single guide RNA libraries, and we do have at hand many validated reagents targeting protein families, as well as genome-wide libraries. So if you're interested, then I would kindly ask you to get in touch with us. But I would now spend also for the remaining time of my talk, I would also like to address an issue that is coming up more and more in the field of genetic screening, which is the field of synthetic lethality or multiplex CRISPR screening. And this is shown here an example. What this means is basically that let's imagine for a moment we have 2 genes in which the knockout of either one, so the knockout of gene A or gene B, does not enforce a phenotype, but when you knock them out or when you interfere with their functionality simultaneously within a single cell, then you establish a phenotype. And why is this important? Well, of course it is important because we would like to understand synthetic lethality, for example, of drug combinations, but also it is very important for pathway redundancy. Many protein families are, and I draw your attention to, for example, the ubiquitin E2 or E3 ligases. There's a lot of redundancy in this pathway, and it would be very interesting to figure out what are the single essential ones, and then what is the next level of combinatorial essential combination. Using the limitation here really is to generate those reagents. And if you look into literature, you will find a handful of papers that used conventional cloning technologies to generate these reagents, and you will quickly realize that the quality of these reagents is nearly what it should be, and which is why we of course now use the 3CS technology that we have at hand in testing whether they work for multiplex CRISPR library generation. And again, this is very similar to the scheme that I showed you before. We basically now have not a single but a double gRNA expressing plasmid, and we use bacteriophages again to extract a single-stranded piece of DNA. We now anneal not a single oligopool but 2, which is highlighted here by the 2 different colors. So now the orange one will become extended towards the green, and the green will be extended towards the orange one, thereby generating a heteroduplex. and also within this process you generate all possible combinations within those individual pools. Then you have the heteroduplex, you transform it into bacteria, and thereby you generate now a combinatorial library. Again, which is the same speed and robustness as for single libraries. And again, of course, I would like to show you this on an example. Here what we did is we tested this on the example again of GFP and mCherry and the combination. What we did here is we took roughly 120 oligonucleotides or gRNAs for GFP and roughly 120 for mCherry. By doing so, we generate 14,000 or 16,000 combinations by simply mixing those two primer pools now into a single tube with single-stranded DNA instead of single pools. And then you wait 2 hours, and then this is basically the reaction product that you see on a gel. Again, I would like to stress that we don't do any PCR or cloning here. And then what we do is we purify the plasmids. And I mentioned to you already before, what we do is extensive quality control of our reagents. And this is shown here by electrophoresis of this combinatorial reagent. I alluded to you before that we are using these homing enzymes or restriction enzyme sites in our templates to make sure that we can get rid of any wild type that bacteria may be unable to degrade. And this is shown here. So when we on the right hand side, of course, the empty backbone will be cleaved with both enzymes because both gRNA expressing cassettes are basically wild type. But if we now introduce the GFP encoding oligonucleotides here, then the ESOI1 site or ISOI1 site is untouched, so the plasmid cannot be linearized, while the SC1 site is linearized. And vice versa, this is for the mCherry pool. Now the other cassette is linearized, while the ISC1 site is untouched. However, most importantly, in the combination of GFP and mCherry, so when you add both primer pools into a single tube, then in the multiplex you don't see any linearization anymore, really suggesting that this site has, that both sides have completely been randomized and changed. So we then of course can also sequence this, and this is shown here. We do quality control of course by NGS, and this is the multiplex combinations of our 2 gRNA expressing cassettes, one having an H7SK promoter, the other one having an HU6 promoter. And this is now really demonstrating the full diversity and also distribution of this combinatorial library. So first of all, it is complete, but it is also very nicely distributed with an AOC of 0.68. Are those reagents functional? That's of course the question here. This is shown on the right hand side. We now took this reagent or reagents and transduced them into GFP mCherry positive expressing cells. And if we do that with the GFP alone virus and then wait a couple of days and then go to the FACS to basically see what is the content of or the percent of fluorescent positive cells, we see with the GFP only library we only target the GFP expressing. And if we do that with mCherry, we of course only reduce mCherry, while in the combination of GFP plus mCherry, we target both genes within a single cell simultaneously, demonstrating or suggesting functionality here. So now this is of course only GFP and mCherry, but I'd also like to share with you now another example that we have. Unfortunately, I will not be able to tell you what exactly the library or the experiment was. Suffice to say, this is an enrichment screen, so we use a fluorescent reporter that enables us to enrich for a certain phenotype. And what we do here is we generate a library, a combinatorial library of a protein family, and we screen this library first on its own. So that means we only target each gene individually. So it's not a combinatorial reagent first, but it's now a single library just targeting those genes, and then seeing which of those genes, when it is deleted, induces the phenotype that we are interested in. We have done this now in duplicates, and this is the correlation again. And we do that to basically identify the single essential genes for that phenotype. This is the correlation, so they're highly correlative. And then we do this with the multiplex library or the combinatorial library, and of course you immediately appreciate the high abundance of dots here. which already tells you that there are much more hits to be expected, but of course also we have more combinations in here. To give you an idea of the size, so the single library on the left hand side has a size of roughly 900 guides, while on the right hand side we have a combinatorial library of about 250,000 combinations. We then, of course, apply computational analysis, and we do outlier analysis to basically perform hit calling. And when we do that, what we see here is that roughly 20 genes, and those are shown here on the left side in blue, have a phenotype in single conditions. But if we now take those single hits and deduct them from all our multiplex or combinatorial hits, we are left with roughly 600 gene-gene interactions, in which none of these genes is essential on their own to induce the phenotype, but only in its combination induce the phenotype. So now this of course demonstrates that these reagents are highly functional and of high quality. And I would like to conclude from this, of course, a few learning objectives here. So first of all, what defines 3CS gene editing reagents? Well, first of all, I hope I have convinced you that there is no sequence bias in these reactions. In these libraries, that it's cloning free, so there will be no cloning needed here. It's very fast, highly reproducible. It's versatile, as it works for CRISPR as well as for shRNA, that I've shown you, and highly reduced cost. And this is of course relevant because we only need a single oligonucleotide per gRNA instead of two. Yeah? Then also which formats are available of single, I mentioned to you, and of course also multiplex formats. And we have validated reagents at hand. And I have another slide that goes more into detail here. But of course, we're also always willing to generate tailored reagents in single or multiplex formats. Validated reagents that we have currently available are, of course, the optimized truly genome-wide library in several different backbones. We have a coding-wide library. Coding-wide library. We also have, those are the validated reagents that I mentioned before to you. Protein families at hand, for example, kinases, phosphatases, tumor suppressors, the entire ubiquitin machinery, autophagy, etc., GPCR, so this list goes on and on. So if you would like to know more about this, then please just contact us and we will be able to communicate with you a full list or either your specific demands. We, of course, also offer tailored and hypothesis-driven reagents in single and multiplex formats, and they come, depending on your needs, with and without a Cas9, and of course also with or without a selection or fluorescent marker. And as I said before, if you have questions on this, then please contact us at info@vivlion.com. So now, of course, we can do these reagents, and some people will ask, okay, what is it good for? And this is why I would also like to share with you some applications for these single and multiplex reagents. Shown here, of course, synthetic interactions can be identified, and this means either gain or loss of function. You can do positive or negative screens depending on the phenotype that you are selecting. Of course, pooled or arrayed. This makes a big difference. Pooled reagents can be easily generated in single as well as multiplex formats, but of course arrayed libraries are easier to generate in single formats than in multiplex because numbers go up very rapidly. Of course, applications are single and multiplex screens. So what can Vivlion do? Of course, we provide validated libraries, as I mentioned before, targeted libraries for a specific set of genes that you're interested in. They are unbiased, and of course we provide them in CRISPR-Cas formats as gRNAs or in RNAi for shRNA. And of course, we would also offer a screen, so in case you don't have the ability to do this, then please approach us and we would be able to communicate this with you. So with this, I hope I have convinced you that we have a very powerful technology at hand that is proprietary to our company and also to the lab, and that we can efficiently generate single and multiplex libraries in various formats, that we have pre-validated libraries, and that we also offer screens. With this, I would like to close. Again, I would like to remind you to contact us if you have questions, and I'd like to thank you for your attention. And my last slide, maybe you can keep this in mind. Vivlion enables basically the next generation gene perturbation. And with that, I thank you very much for your attention, and I'm happy to take questions.